This lecture is about the mechanisms by which drugs can bind to and block ion channels. They can block in different ways, with different consequences for cell function. If you're watching this video, you probably already know a bit about ion channels. These are proteins that span cell membranes and contain a central pore. When the pore is open, ions can flow through the channel from one side of the membrane to the other. This ion flow changes the membrane potential and influences any cellular process that depends on it. Ion channels can open or close to regulate ion flow depending on the surrounding conditions. Some channels respond to changes in voltage, while others open when a specific ligand binds to a receptor site on the channel protein. Regardless of how a channel opens, it can be blocked by drugs. In this video, I'll discuss the general mechanisms by which drugs block ion channels. So what I explain will apply broadly no matter how the channels are normally gated. To understand the different types of drug blockade, you first must realize that ion channel proteins can take on different shapes or conformations that can affect drug binding. Under resting conditions in a cell, most ion channels are in a resting or closed state. The gate that controls ion flow is closed so that ions cannot access the pore and cross the membrane. When exposed to an appropriate stimulus, like membrane depolarization or receptor agonist, the gate opens, allowing the free flow of ions across the membrane down their electrochemical gradients. This is the open or activated state. During a maintained stimulus, many ion channels gradually morph from the open state into a non-conducting state, as a different part of the channel creates an obstruction. This is known as an inactivated, refractory or desensitized state. Although the pore is still open, ions can no longer get through it to traverse the membrane. Drugs can block ion channels by interacting with them when they are in any one of these three states. They can directly obstruct the channel pore, preventing ion flow through the channel. Some drugs bind to allosteric sites that cause conformational changes affecting the channel's ability to open or close. Other drugs interact with the channel's gating mechanisms to alter its ability to open and close. Pore blockade, or open channel block, occurs when a drug molecule enters the channel pore while it is open and binds to a specific site within the channel. While the channel remains open, the drug can bind and unbind dynamically, depending on its reaction rate constants. While the drug is bound in the pore, ion flow is blocked. Once the drug leaves its binding site, the channel gate can close. This closure hides the binding site, preventing the drug from exerting its effect. Most open channel blockers work this way. However, some drugs bind to sites deeper within the pore. If the drug is small enough, the channel gate can close even with the drug still inside. In this case, the drug becomes trapped within the channel and cannot unbind or exit until the channel reopens. To illustrate the consequences of open channel block, these traces represent ion channel currents recorded from a cell under voltage clamp conditions in response to activation by a brief stimulus, indicated by the arrowheads. If you're not familiar with the voltage clamp technique, see my video on that topic. Now let's see what happens when we add a pore blocker and repeat the same stimulation. The first thing to notice is that the peak of the current is unchanged. The main effect of the blocker is a faster decay or recovery of the current after the peak. I've superimposed the original trace as a dotted line so you can clearly see the difference. This happens because the drug has no effect until the channels open at the peak of the response. Also, because the stimuli are delivered at a low frequency, there's enough time between pulses for the drug to unbind and leave the pore. So block only occurs during the currents when the channels are open. Now let's see what happens when we increase the frequency of stimulation. This time, when the pore blocker is added, the peak amplitude of the current gradually decreases with each stimulus until it stabilizes at a lower plateau. This occurs because more frequent channel openings increase the likelihood that the drug will enter the pore and bind. At the same time, there's less time between stimuli for the drug to unbind and leave the pore, so the block accumulates in more and more channels. If the stimulation rate is then reduced back to the original low frequency, the current rapidly recovers. That's because the longer interval between stimuli now gives the drug enough time to dissociate from the channels. Things look a bit different when the pore blocker becomes trapped inside the channel. During high-frequency stimulation, the drug causes accumulating block just as it does with blockers that are not trapped. The difference becomes clear when we switch to low-frequency stimulation. This time, there's no recovery between stimuli because the channels remained closed and the trapped drug could not escape. However, if a second stimulus is applied after a longer delay, some recovery is observed. That's because channels that briefly opened during the earlier stimulation allowed some of the trapped drug to unbind and exit the pore. Allosteric sites on ion channels are regions outside the pore where drug binding induces a conformational change in the channel protein. This typically leads to channel closure or a reduced ability to open. Allosteric sites located on the extracellular domains are accessible to hydrophilic or charged molecules acting from outside the cell. Sites on intracellular domains respond to drugs that must cross the cell membrane to act from within. Some highly lipophilic drugs can partition into the lipid bilayer and access transmembrane allosteric sites. Drugs acting through allosteric sites commonly stabilize the channel in either the closed or inactivated state, thereby reducing the number of open channels and the ionic current. Importantly, different drugs preferentially bind to and stabilize different non-conducting states. Some target the closed state, while others selectively stabilize the inactivated state. This diagram shows the transitions between the closed and open states of an ion channel. Under resting conditions, channels are mostly in the closed state because the rate of opening is very low compared to the rate of closing. When a stimulus is applied, it increases the forward rate, shifting more channels from the closed to the open state. Once open, ion channels often undergo inactivation, either quickly or slowly depending on the channel type. Inactivation is a one-way transition. Channels in this state remain non-conducting until the stimulus is removed, allowing them to return to the closed state during recovery. Now let's add a drug that acts as an allosteric blocker, which preferentially binds to the closed state. In this case, closed channels are drawn into a drug-bound conformation from which they cannot open. This means fewer channels are available for activation by the stimulus. However, channels that are not drug-bound continue to open and inactivate normally. When we record membrane currents in response to low-frequency stimulation, adding the drug causes a reduction in peak current amplitude. Although not shown here, This inhibition increases with rising drug concentration. With high-frequency stimulation, the drug produces a similar reduction in current amplitude. Notably, the extent of inhibition is the same regardless of stimulation frequency. This pattern clearly distinguishes the action of a closed channel blocker from an open channel blocker. Now, let's add a drug that acts as an allosteric blocker that preferentially binds to the channel in its inactivated state. Binding occurs only after the channel has opened and transitioned into the inactivated state. The drug then stabilizes the channel in a drug-bound inactivated conformation from which it cannot return to the closed activatable state. As a result, the pool of closed channels available for activation is reduced, while channels not bound by the drug continue to open and inactivate as usual. When membrane currents are evoked by low-frequency stimulation, the drug has little effect. That's because channels spend very little time in the inactivated state. They open briefly and may only inactivate transiently, if at all, leaving minimal opportunity for drug binding. In contrast with high-frequency stimulation, the drug causes a cumulative block, similar to what is seen with open channel blockers. This is because more frequent channel openings lead to greater inactivation, providing more opportunities for the drug to bind. Drugs that block inactivated channels have another important and clinically useful property, which I'll now demonstrate. In this example, a small but sustained stimulus is applied before and during low-frequency stimulation. After a few pulses, the repetitive stimulation is paused and the drug is applied. Stimulation then resumes at the same low frequency. You'll notice that the peak amplitudes of the membrane currents are reduced, indicating that the drug has accessed its inactivation-specific binding site and is now blocking the channel. The key point is that the sustained low-level stimulation leads to a small but persistent population of channels opening, which in turn results in inactivation. This increased time spent in the inactivated state provides the drug with an opportunity to bind and cause channel block, even without high-frequency stimulation. Use dependence is a term often used in association with ion channel blocking drugs. It is the functional outcome of frequency and state-dependent block, and simply means that the drug's effect increases with repeated or sustained use of the channel. In other words, the more a channel is activated, the stronger the block becomes. This is a key property of drugs that bind preferentially to the open or inactivated states of the channel. From a therapeutic perspective, use dependence explains why certain drugs selectively block overactive or depolarized cells, for example, in ischemic or arrhythmic tissues or in rapidly firing neurons.